we're going to talk about the role of genetics in neuromuscular disease. And as you probably know, neuromuscular disease is not a single condition. It's an umbrella term, and it's a group of disorders characterized by impairment of one of these three areas, either the muscle fibers themselves, which is this red down here, this tube-like thing, or there can be impairment of the nerve cells that control the muscles, and so that's this area in here. The third area that can be affected, that can cause a neuromuscular disease, is the junction, or the space, between the nerve and the muscle. So this is this neuromuscular junction right there. So any of those areas can be affected and cause a neuromuscular disease. So if the muscle fibers themselves are affected, you're going to have abnormal function of the muscle, degeneration of the muscle, and some examples would be myotonic dystrophy and Duchenne muscular dystrophy. If the nerves are altered, the nerves could be altered in the peripheral, meaning the limbs. An example of those kind of disorders would be Charcot-Marie-Tooth. If the motor neurons, which are in the spinal column, are affected, an example of disorders that result would be spinal muscular atrophy, or SMA. In that neuromuscular junction, if there's an issue there, genetically speaking, an issue that can arise is called congenital myasthenia syndrome. There are over 150 neuromuscular disorders, and there are over 400 genes associated with neuromuscular disease. But as we heard in our last talk, not all neuromuscular disease are genetic. Some are autoimmune. So when we establish or try to establish a diagnosis of neuromuscular disease, you all know that there's got to be a neurological exam and evaluation, and that evaluation can be pretty extensive. It can involve multiple series of tests, and some are frankly invasive, like a muscle biopsy. And there's usually blood work, and one of the biochemical levels we like to measure is called creatine kinase, and that gives us an idea of muscle degradation. Certainly, if there's a positive family history, knowing that can help the clinician because it sort of pinpoints what disease is already in the family, and then we can decide, hey, is the symptoms presenting in front of us related to that family history? And then now we have the option of genetic testing to identify a pathogenic variant. Pathogenic just means disease-associated or disease-causing. And so that's where we're going to focus our talk today. So the outline of our presentation today is going to be discussing the utility of genetic testing in neuromuscular disease, focusing on three areas. And the most obvious is confirming the clinical symptoms. So a patient presents to the doctor's office with a set of signs and symptoms. We want to try to use genetic testing to come up with that diagnosis. Well, why? We'd like to avoid unnecessary, expensive, and even invasive additional testing like a muscle biopsy if we can. We'd also, for many families, it's been a long time since they're trying to get to an actual diagnosis. So if we can end or shorten that diagnostic odyssey with a simple blood test, for instance, we'd want to do that for the family. And then once a genetic diagnosis is confirmed, that gives us an idea of perhaps severity, disease course, progression, and helps the family understand what things perhaps will be like in 5, 10, 15 years. The second aspect that we'll discuss today with genetic testing is to understand then the inheritance of the particular neuromuscular disease, then be able to predict what other family members could be at risk, and whether or not there's a risk for future children to also have the same disorder. And then thirdly, as was discussed in our last talk, I'll touch very briefly on the benefit of genetic testing in trying to tailor certain treatment or medical management. And of course, the ultimate goal, we're not there yet, but the ultimate goal is to be able to prevent. So before we go any further, I'm not going to go a whole lot of detail here, so this is just an overview of genetics primer. So we have DNA, about 7-mile-long strand of DNA in every cell, and segments of DNA are broken up into segments we call genes. And they're the instruction manuals. That contains data, and that data produces proteins, and these proteins are used in normal bodily function, development, etc. The data in the gene is encoded in a 4-letter alphabet. The English alphabet has 26 letters. DNA alphabet has 4, A's and T's and C's and G's. And the sequence of those alphabets, or we call them nucleotides, the sequence of that's our instruction manual. The DNA, like I said, 7 miles long, so it's got to be compacted some way, like a spool of yarn or thread wrapped around itself very tightly. So it's wound or packaged really tightly into a structure called a chromosome, and those chromosomes are contained in the nucleus of our cell. So humans generally have 46 chromosomes. They come in pairs because we get one copy of a pair from our mom, one copy of the other copy from dad, and so forth. The first 22 pairs are simply numbered, and they're called autosomes. And they're the same whether you're male or female. The 23rd pair are the sex chromosomes, and of course they're going to differ depending on whether you're male or female. And females have 2 X chromosomes, and a male has an X and a Y. I want to show you here just a pair of chromosomes. This is chromosome number 5. Just as we have 2 copies of every chromosome, we then have 2 copies of every gene contained on that chromosome. So for instance, if the blue chromosome came from dad, the pink one came from mom, let's say we're looking at gene D, we have a copy of gene D we got on the chromosome from mom, but we have another copy of chromosome D that we got from our father's chromosome 5. Does that make sense? Okay. Now the caveat to that are the genes located on the X chromosome, because a male only has 1 X chromosome, so therefore he only has 1 copy of the gene on that chromosome. Whereas a female, we would have 2 copies of the gene that's located on the X chromosome. Why? Because we females have 2 copies of the X chromosome. All right. The other thing to keep in mind with this X-linked stuff is if we do genetic testing and we find a gene with a variant in it, and it's located on the X chromosome, that's important for us to know because that gives us an idea of, well, why is this affecting males more than females? And that it helps us understand inheritance patterns. So let's speak briefly about the process of genetic testing. I've listed a few of the laboratories on the, well, you're right, that we use for neuromuscular disease genetic testing. There are others. This is not an all-inclusive list. But if a patient presents with symptoms, or there's a family history of a very specific disease, like the child's maternal uncle has Duchenne's muscular dystrophy, well, then the clinician is very rightly going to say, let's just order the genetic test that looks at the gene dystrophin because I know that gene causes muscular dystrophy. So we'd very simply order a single gene test, just looking at that gene. On the other hand, if the symptoms are pretty vague or common, and there's no family history, or there are multiple genes responsible for a single type of condition like Charcot-Marie-Tooth, well, then we're going to want to order a multi-gene panel. And that's exactly what it means. It's a single test that can have anywhere from 2 to over 100 genes on one test. So instead of, for Charcot-Marie-Tooth, for instance, instead of sequencing each gene individually, let's just do it all at once on this panel. And results usually come back in about 2 to 4 weeks. Lastly, if all this targeted testing fails, as we heard in our last lecture, whole exome sequencing is clinically available, and it sequences the coding or the protein coding regions of all of our 20,000 genes. Now, we don't typically start with that test due to several factors, but number one being cost. We can also find a lot of variants of uncertain significance, so we start more targeted, and then if need be, and if the patient desires, we might be able to do a more extensive look in the genome for a genetic cause. Once genetic testing is performed, there are generally 3 types of results that we can get back. The first is pretty obvious, a normal or a negative result, And that simply means that there was no genetic variant found in the gene or the genes that we were looking at. Now, that doesn't mean there's not a genetic problem going on. It's just that in 2018, we weren't able to find it or identify it yet. If we get a positive result, that's pretty clear as well. That means we did find a genetic variant, and we know that that is responsible for the condition, and we can then go forward. The third possible result that we can get back that I like to make sure my families understand before we even do the testing are variants of uncertain significance. We call those VUS for short, and they mean just what they sound like. These are genetic changes or variants in the gene that either we don't know if it's associated with disease, or we've never seen it before, or sometimes these are variants that might be present in me and just make me different from you, not necessarily disease-causing, because we all have variants in our genome. An example, let's look at the word gray. I might have an A, you might spell your word gray with an E. Is that problematic? Well, no, I still read that word as being gray. It just makes me different from you, but it's not disease-causing. So when we do genetic testing, it's important that families understand that clearly we might get a very clear, obvious negative or positive, but a lot of times we do get variants of uncertain significance. Now, the benefit with genetic testing is that over time, as more people are getting tested, these variants of uncertain significance may lose their insignificance. We may be able to classify them as just benign, routine variation between me and you, or no, we may learn that, yeah, that change is problematic, and it is in fact now associated with that condition. So as time goes by, we learn more about these VUSs, and they do get reclassified. So back to our outline about what we're talking about today. So the first use of genetic testing is, again, to confirm a diagnosis. Again, I've shown there's numerous neuromuscular diseases, and many present with similar signs and symptoms such as poor balance, frequent falls, difficulty walking, progressive muscle weakness and wasting. So when we have this sort of broad symptomatology that could be caused by any number of things, it might be nice, can we narrow that down in some way? The age of onset of symptoms can vary, even within the same condition. So if we have a woman that presents with myotonic dystrophy, and she says, well, I was diagnosed in my late teens, but my newborn here's got issues, I don't think it's the same thing because I was diagnosed later. Well, no. The same disease can simply present at different times in different generations. So when we do genetic testing, it can be very helpful in pinpointing the exact diagnosis, especially when the clinical picture can be so varied. For many families, before a diagnosis is made, they report being on a diagnostic odyssey. And this is the period when caregivers or parents first become concerned about their loved one until the time of a diagnosis actually being made. And this is a time, especially with children with neuromuscular disease, this period can be a few months or even a few years. It's a time of uncertainty and emotional turmoil for families, and it's often characterized by multiple doctor visits, minor medical procedures like those listed here, and many of these can be painful and even frightening for young kiddos. So if we can start with a test like saliva, a cheek swab, and get our answer before having to do all of these more invasive tests, that can save the family and the child some of these other testings. A few years ago, there was a published study of interviews of 10 moms regarding the impact of this diagnostic odyssey on their children and their families. Once the diagnosis was made, the interviewees reported that they felt validated in their initial concerns. One mother was quoted to say, "To hear the words, this was a scary moment, but in a sense, it's going to sound bad, it was so confirming to me. I knew something wasn't right." Another mother reported that the diagnosis provided proof that their child wasn't being lazy in gym class. There was actually a reason the child wasn't able to keep up and do what the other kids were doing, because a diagnosis was now made, and it again confirmed their initial concerns. A genetic diagnosis also provided prognosis. The parents could prepare for the progression of the disease course, and as age appropriate for the child, could help the child understand what might be coming down the road, for instance, the use of a walker or the eventual use of a wheelchair. Additionally, some families felt a sense of relief when they got a diagnosis because they were expecting a more significant or perhaps life-threatening disease, to then be told the disease that their child actually has is not as bad as they initially were fearing. And lastly, families were able to tap into established support groups and networks once the diagnosis was made. So moving on to the second role of genetic testing in neuromuscular disease, and this is predicting familial risks. If a genetic diagnosis is made, of course we can then know what that gene is, know what the condition is, but now we can then determine what other family members are at risk, are other children at risk, and are future children at risk. The condition may have occurred for the first time in the affected individual, and we call that sporadic or de novo. A lot of times we're going to need parental studies to help confirm that. We're going to need to test the parents to see if the parents have the gene. If the parents do, then that gives us a clue, yes, this is inherited. It's important to remember that even if the altered gene is de novo or occurred for the first time in that person has that gene change, and that person can now pass it on to his or her offspring. The disease-causing genes can be inherited generally in one of three ways, and we're going to review these now. So the first is autosomal dominant. If you remember back to when we looked at the chromosomes, that word autosome means that the gene is on one of those numbered chromosomes, so these kind of conditions occur in males or females. And dominant simply means that you only need one altered copy of the gene to express disease. The altered gene can be, again, inherited or simply occurring for the first time in the affected person. So in this case, we have a dad with the condition. He has one gene that's not working, one gene that is. And when he has children, he has a 50-50 chance of passing the working or the non-working gene on. And it doesn't matter if his child's male or female because, again, the gene is on one of those numbered chromosomes. The other type of inheritance pattern is autosomal recessive. So again, the gene's on one of the numbered chromosomes, so these disorders can affect male and females equally. But recessive means that in order to express disease, both copies of that gene have to be altered. And so usually what happens, so let me just show you here, this little guy here, he's got no good copy, no working copy, so he's affected with the condition. And generally speaking, the child inherits an altered gene from mom and an altered gene from dad. Parents are not affected generally because they have a working copy of the gene as well. So if there are two carrier parents of an altered gene that's recessively inherited, each time they have a child, there's a 1 in 4 chance or 25% chance that a child would be affected. The third inheritance pattern is X-linked recessive. So this means the gene is located on the X chromosome. Usually affects males because, again, they only have one X chromosome. And in about 2/3 of cases that are caused by an X-linked gene, mom is a carrier of that gene on one of her X chromosomes. And so that's what's illustrated here. Mom is a carrier of a non-working gene on her X, but on her other X works fine. So she has a 50-50 chance whether she passes the working X or the non-working X on. If her son inherits the non-working copy of the X, the son would be affected. Now, you may know, for instance, with Duchenne muscular dystrophy, female carriers can express some signs and symptoms as well. So we can't say with 100% certainty that women who have an X-linked recessive gene will have no problems themselves. And knowing that they carry that allows us to do some screening and make some management recommendations for those women. So when we test a child and the child has an X-linked condition, we do recommend testing the mom, not only for recurrence risk, but maybe we need to recommend some medical management or screening for her as well. So having a diagnosis genetically confirmed not only confirms that diagnosis, but allows us to make some prediction about future children and whether other people in the family, we didn't go out to aunts, uncles, cousins, nieces, nephews, but knowing the inheritance then allows us to figure out who else could carry this gene if it's recessive. What other children, if any, should be tested, and when should they be tested? So lastly, the third role of genetic testing for neuromuscular diseases is in aiding more appropriate treatment and advancing treatment options. For most neuromuscular diseases, the treatment and management is supportive, as we've discussed earlier, and it's based on symptoms. But the goal is to increase lifespan, improve quality of life by focusing on treating or delaying those symptoms, enhancing physical mobility and the social interactions of individuals with their peers, and then certainly to try to prevent more life-limiting aspects of the disease like heart and lung complications. The addition of a genetic diagnosis may aid in research, as we just discussed in our last talk, and eventually the enrollment into clinical trials for various treatments or therapies that are disease-specific, but we can go even deeper than that. We're doing clinical trials, as was discussed before, that are gene-specific, and we can go even deeper than that, variant-specific. Depending on the misspelling in the gene, there might be a specific therapy available. So let me very simplistically explain how this works. So here's our chromosome. We know that where that working gene is. That working gene makes a working protein, and that working protein helps aid in normal nerve or muscle function. So that's the good. So down here we have our chromosome. We have a gene that's not working like it's supposed to, produces an altered protein, and therefore it has a bad effect on the nerve or the muscle cell that it's supposed to work on. So once we know the course of this, can we somehow come here and affect the gene directly or indirectly affect how the protein works on the nerve or muscle cell? So that's just a Google Earth view of trying to understand the genetics so that we can figure out where can we intervene and stop disease or limit this progression of disease. So here are some examples of some molecular therapies that are currently clinically available for 3 neuromuscular diseases. The first 2 help the altered gene make more functioning protein in our body. So Exondys received FDA approval in September of 2016 for use in a subset of patients with Duchenne muscular dystrophy. In our last talk, that was discussed in a bit more detail, and I won't go over all of that. But again, it's just a subset of people with Duchenne's based on their genetic test result, the specific variant they have. And more therapies are in the works to try to make it broadly available to other children with Duchenne's muscular dystrophy, again, based on their genetic change. Spinraza received FDA approval in December of 2016 for use in all patients with a genetically confirmed diagnosis of SMA. So again, in order to receive this treatment, we have to have confirmation genetically that it is SMA, because what this treatment is doing is allowing the body to produce more of that SMN1 protein that is lacking in someone with SMA. So we have to make sure that's the condition they have, so we're targeting and treating the right problem. Now, unlike the other 2 therapies that help the body produce more of its own protein, Lumazyme is a medication or a therapy that replaces a missing protein with a synthetic one. So we're giving the body a protein that it's not able to make on its own. And Lumazyme received FDA approval in May of 2010 to treat adult-onset Pompe disease. But then in August of 2014, Lumazyme received an extended FDA approval for use in patients of all ages with Pompe disease. So because of these new molecular therapies, and certainly new ones on the horizon, there's a push for earlier diagnosis via newborn screening. Because if we can start treatment early, the goal is to perhaps slow or prevent progression of disease. So Pompe disease was added to the National Newborn Screening List in 2013, and today many states do incorporate that in newborn screening. Spinal muscular atrophy is the latest neuromuscular disease added to the National Newborn Screening List, and currently there are 6 states that have incorporated SMA to their newborn screening panels. 4 additional states have started pilot programs. Here in Pennsylvania, the Department of Health is currently and actively working on creating a program to incorporate SMA into our newborn screening program as well. As far as Duchenne muscular dystrophy, again, there's a push to get that on the National Newborn Screening List. It's not there yet, but the MDA and other stakeholders are currently advocating for its inclusion, because once it gets on the national list, a lot of states say, oh, well, maybe we ought to put it on our state list. And so that's the process that happens. So in summary, in closing, in my mind, the role of genetic testing in neuromuscular disease is threefold. 1, confirm a diagnosis, hopefully quickly, without having to do a lot of extra and sometimes invasive tests. And hopefully end the Diagnostic Odyssey, or shorten it. Provide families with risk estimates for future children, or determine who else might be at risk for the condition. And then thirdly, the role of genetic testing can aid in better treatment or better management options. So that's the talk. Do you have any questions?
We would like to
That's.
be provided a copy of your presentation.
Will there be a copy of this? Okay. Yes, sir.
I just want to ask you, with all the muscular diseases out there, would you know what Beckers would be closest to? Is it Duchenne?
Beckers Muscular Dystrophy?
Yeah.
Yes, so it's a problem in the dystrophin gene. So it's sort of, we kind of think of it as a milder form of the Duchenne's. So it's X-linked. Mm-hmm.
Oh, okay. And like with some of the same therapies for Duchenne eventually work for?
My understanding at this time, we don't, and maybe Doctor, you could help me, but I don't believe there's therapy specific for Becker.
Actually, an allelic disorder where people with Beckers make some dystrophin, whereas people with Duchenne make no dystrophin. And so that small amount of dystrophin is partially functional, and that's why it's typically milder. There have been some studies looking at the Flazacort
Okay.
with Beckers, some indication that that might be useful. And certainly, depending, because you make some dystrophin, some of these exon skipping may or may not be, nobody's applied them to Beckers yet. In theory, could you make somebody make more dystrophin with an exon skipping treatment? Nobody's actually examined what the effect would be if somebody's already making some. So that's why they haven't been applied, but it is the same gene, and the difference is that when somebody has Duchenne, the gene is so bollocksed up that you make a protein that's so abnormal it's rapidly broken down, or doesn't even get made at all, whereas you have some protein, so it's not clear that enhancing it will help you as much as some of the other strategies. In fact, one of the treatments for Duchenne has been looked at is to take somebody with Beckers and model a gene therapy after the mini gene of a mildly infected Beckers, so that mildly infected Beckers has a smaller protein that could be encapsulated in the virus and given to a patient, and at least create a situation where somebody has partially functional dystrophin. So it's a long-winded way of saying that we have applied some of the treatments. It is really the same disease, but different molecular sort of biochemistry, that you at least have some protein.
Yeah, I was diagnosed when I was 19, so my mom's 66.
That's
I
right.
got through, and I had a lot of muscle pain during this course.
Yeah, that's one
Right.
of the phenotypes, is actually muscle cramps, and others just having a heart problem. And then there's people who
mutation, the TFG.
Oh, sorry,
TFG.
Okay.
And so my daughter actually, 19 months, it was originally done by a centronuclear myopathy from a muscle biopsy. Long story short,
Yeah.
it falls somewhere between the symptoms of like SMA, ALS, but it's extremely rare. And because of that, I'm wondering what kind of clinical, there are no clinical trials. Like, is anything ever tested off, like saying this is what it sort of follows? So even though you don't exactly have this, they will still trial for those extremely rare cases.
I would think the answer is no, because the clinical trials are geared to a specific problem, a specific gene, a specific variant, because they want to show efficacy for that.
Right.
They don't want to have they don't want to go down rabbit trails.
Right.
They want to stick and show what that.
So for people that have that extremely, very rare, like youngest diagnosis that's researched is age 20. My daughter was 19 months.
Yeah.
So, I mean, it's just very, very rare.
I'm sorry.
And so there's no, okay. I just wondered whether they ever trial for anything that shows similar symptoms.
Do you have any?
There are trials, the ones, but they're not going to be specific for the
Right.
genetic defect.
Right.
Okay, so I have two comments. One is that as we get better and we understand CRISPR and we understand gene replacement therapies, we can apply those tools to any disease.
Mhm.
So that's number one.
Mhm.
Number two, there are trials that are sort of general for neuromuscular
Mhm.
disease and are applicable to anyone with weakness, because the goal is not to treat a specific defect, but to enhance muscle strength or muscle bulk. And so some of those, like that ACE-083, is
Mhm.
being applied to, quote, neuromuscular disease in general. So
We just need to look broader.
Yeah,
Yeah.
well,
Zoom out.
Yeah, we identified it, but we need to zoom out.
and there's also some other skeletal muscle activator trials going on that may be more broadly applicable once those drugs get through FDA. People will use them off-label because the mechanism.
Okay.
Go ahead, Sydney.
Does it really matter what company you use for the genetic testing? My son was diagnosed here at Hershey, but then you see all these other companies, and what really is the difference in what they're doing and the efficacy of how well they're doing it?
Did you receive a diagnosis? A genetic change was found? So that's it.
And we had the muscle biopsy.
Okay, yeah. So that's it. So it's not like one result from one lab is better or worse. So I've got a diagnosis. Do I really believe that? Well, let me try lab 2. No, you don't need to go that route. A lot of it comes from insurance and maybe some technique in how the lab does its job in the laboratory, the sequencing and so forth. But that's up to the clinician, the genetics people, to determine what laboratory might be best to use. Yeah, but if you've got the answer, you don't have to mistrust that and say, well, let me see if lab 2 can get the same thing or something different. Yeah. Go ahead.
I have a question about the genetic testing. So the genetic testing they're doing now is being done in newborns? So once they're born, so it's not at the 16 weeks that they do for when you're pregnant, correct?
Well, if you're talking about newborn screening,
Mhm.
that's like, when they prick the baby's
Yeah.
heel, okay? So it's not looking at all kinds of neuromuscular disease. We're just focusing on concern for Pompe, SMA at this point.
Okay, but they're doing that with the newborn, not, because they do, like, I was told at 16 weeks that my daughter would be either a Down syndrome baby or have spina bifida.
Okay.
So we did, the
amnio.
The amnio and the whole bit, and they came back and told me everything was going to be fine.
Okay?
And then 12 months, I was told I was an overreactive mother because my daughter was not walking. And
Okay.
at 15 months, they decided she had SMA.
Okay.
So,
So in pregnant moms or individuals who are considering pregnancy, we have what's called carrier screening, which is where we offer families the opportunity to screen themselves. So the mom, dad, prospective mom and dad, to see if they are carrying certain recessive disorders.
so when her older sister decides to have children, she can be screened?
Yes.
Okay.
And again, the purpose there is to know, are you carrying genes that could be passed on? Now, it's not telling you the child that you're going to carry will have a disease. Then you're going to have to do an amnio, or we can go down the whole other in vitro fertilization route and that sort of thing.
Right.
But we just don't test babies, fetuses, to see if they're going to have this disease or that disease. we need to know what disease we're looking for.
Now, if you're in a state like Pennsylvania that doesn't currently do that in their
Doesn't
newborn
do
screening,
SMA,
can
right.
you ask if you know you're a carrier?
Oh, yeah, oh, that's a whole different ball of wax. Yes, yes. So if you know that you're carrying a genetic disease, it's not gonna be part of the newborn screening panel. You're gonna say, hey, my kid's got a 1 in 4 chance to have this disease. We're gonna do genetic testing immediately. Yeah, it's just not part of the national newborn screen. That's done on everybody.
All right,
It's gonna be specific to your family.
okay.
Yeah.
Thank you.
Mm-hmm. Yeah.
Yeah, in 1966, I had a muscle biopsy,
Okay.
and they said I had pseudohypertrophic muscular dystrophy.
Okay.
But now, when I go to the neurologist, they say, oh, we don't use that term anymore.
Okay.
And I've done genetic testing at the NIH and at Hopkins, and right now I'm in a position where they're saying that my aniline is a little high, and I've sequenced my entire mitochondria on my own.
Okay.
I guess over 15 years ago, and we just use it for genetic genealogy
Okay.
purposes, because in Maryland, you can't test for medical conditions. So I went up to Corelli, and Corelli got me in contact with the NIH before they really started to do the studies. But basically, they were saying, at least the Air Force Academy was saying, that the pseudohypertrophic muscular dystrophy was associated with vitamin deficiencies.
Okay.
And they still want to do another muscle biopsy because Kernan's lost the original results. But, you know, that takes a long time. And what I'm a little confused about is I haven't heard a lot about mitochondrial types of studies with the Krebs acid cycles, you know, dystrophies that are mitochondria,
Yeah.
associated. And as I said before, the NIH and Hopkins, they're still uncertain because they haven't found a positive result. And I guess I listen to the doctors, and most people say that we're changing the definitions now. Just like in genetic genealogy, most of the sequences that we have done aren't classified. And I actually had to send the data to NIH, and there wasn't any mutations reported. These were the first type of mutations. And I guess I'm a little confused because my gait's starting to stiffen, and earlier in the lecture we talked about the motor disorder. And at first, of course, they actually looked at my calf muscles, and I didn't have Duchenne. It's more like some type of limb-girdle type. But I don't know. I've heard over the last few years that a lot of people haven't been diagnosed.
Oh, well, that's for sure. That's for sure. I mean, what we know now in 2018 won't be what we know in 2020. So if genetic testing is done and it's negative, it doesn't mean, like as I said, that there's not a genetic cause. Obviously, there can be other causes, environmental causes, vitamin deficiency, et cetera, but those can be ruled out in other ways and other tests by your clinician. But genetic testing may be revisited by your clinicians 5 years from now because new knowledge is available.
Well, but they asked for blood samples for my two sisters.
Yep, makes sense. To just see what variations maybe are different from you than them, and trying to hone in on the genetic change that's causing your disease. Yeah.
But is it true that no one uses that term pseudohypertrophic muscular
I'm not I don't know. Doctor, do you know?
It would be very unusual, because it doesn't mean much.
Oh, it doesn't mean much?
It's sort of a,
It's a broad term. It's just describing,
It's just a broad term?
it's just a description.
we
that was
used a lot of newer terms.
Yeah, newer term.
Unfortunately, even from when I started training, the lingo has changed quite a bit. And it was in use years ago, but now, we typically like to figure out which genetic had or not. And limb-girdles,
Sir, did you have a question?
Yeah, you mentioned about VUS variant of
Uncertain
uncertain significance. What does it mean when the report says it's a variant of uncertain significance associated with the disease? One of the diagnoses my daughter had, I remember reading it in the report that said it's uncertain significance associated with Beckers Muscular.
Okay, so I think what they're saying is the gene that the variant was found in, the gene itself is known to be associated with Bethlem myopathy, but the misspelling in the gene that your daughter had, they've either never seen it before, so again, it's like that gray and gray. The gene itself has been associated with Bethlem, but the change in the gene, they're not sure, does it cause the protein to malfunction or not? So I think perhaps, without obviously seeing the report, I'm thinking that's what it is stating. The gene has been associated with the condition, but they found a change in the gene that they're not sure if it actually causes the gene to malfunction, and so it might not be disease-causing. Does that make sense? Okay. Yeah.
Quick question. I've been diagnosed with something very generic so far, I found.
Oh, boy.
Being distal myopathy, which is a big, broad.
Sure, yeah.
And I'm wondering if, I haven't had any genetic testing done, but I'm wondering whether at this point in time, and I've had probably 10 years worth of this, at this point in time, there might be something that would, genetic testing-wise, would bring it out of that broad category, narrow it somewhat.
It could, I mean, it's certainly worth a try to speak to your doctors about doing a broad, or what we call comprehensive neuromuscular disease panel, or if they want to, so these panels can be, like I said, hundreds of genes, or if they kind of know what type of, if it's a myopathy or some other issue, they might be able to narrow down the panel. But yes, if your goal is to try to come up with an underlying etiology, it's certainly something to speak to your clinician about. I hate to say it, but there's always the elephant in the room of, will insurance cover it?
Yeah, it sounds like you're getting outside of the realm of insurance coverage.
At this point, they're probably going to say, what's it going to matter? I mean, we have kids that we try to get tested, and they're like, is it going to change their management? We're like, well, it might. They'll argue with us. So in a gentleman of your age, they might say, look, it's not going to change a thing. So I don't know.
Basically, I'm not looking at it from you, but from
Correct.
you.
I hear you. I agree with you.
Asking me, do you know whether?
Exactly.
I can't answer.
And so we all agree clinically that that testing would be great, and it would be lovely if your insurance paid for it. Just upfront and honest, they may have a different take. So it might be, and again, this is where your question, ma'am, earlier about what lab to use. Well, it may come down to, honestly, to pricing. Who's got a better self-pay price?
There are some panels that are available, and your doctor should know where, if your insurance doesn't cover, your panels, depending on your clinical presentation.
Mm-hmm.
Panels for the limb-girdle dystrophies, for instance, it can be done at no cost to you. First you have to do investigation of insurance. But there are some that we can get,
Yeah.
SMA testing sometimes get done for free. And
Yeah.
the Charcot-Marie tooth testing vis-a-vis amyloid testing. So
Yeah.
there are some ways, depending
Mhm.
if you talk to your doctor, if you really want to know.
Yeah.
Is the topic of gene counseling something that is done specifically, I've got this, but is that done also with the doctor?
So, yeah, I mean, your doctor, if they're comfortable talking about genetic testing and all of that, then yes, they can serve as your genetic counselor. Otherwise, they may refer you to a specific specialty, me, that is the genetic counselor. So it doesn't have to be a genetic counselor. It can be a physician who's comfortable doing it.
Does the MDA have a list of specific doctors who know more about muscular dystrophy than other neurologists?
Yeah, I would assume do you have, well, they have a list of MDA centers, right?
Right, yeah.
So in order to be an MDA center, there's got to be a level of competency and
So contacting the center would be able to give me
yes.
information.
Yes, yes, yes. How are we on time? I don't,
Yeah.
I mean, I'm happy to stay, but I also understand.
Yeah, well, first of all, thank you, Melissa.
Yeah.
I think she,
Thank you, thank you.